exacts its cost. Well, some experiments have been done now, and it turns out that's not true. Aging exacts a huge cost in the wild, maybe 20, 30, sometimes 50, 70% of fitness in experiments on flies, in surveys of mammals. So, again, it can't be an accident. There is a lot of evolutionary pressure around aging that couldn't have been recognized in 1952. Point 4 is really technical. If you're an evolutionary biologist, you know about additive genetic variance. It's a measure invented by Fisher that measures exactly whether something is an accident of nature or whether it's an adaptation. If additive genetic variance is low, that means it's an adaptation. If it's additive genetic variance is high, that means, well, nature doesn't care about this very much, just letting the animal drift. And there have been studies of additive genetic variance in flies, very detailed studies, very well-designed studies, that show that not only is the additive genetic variance for aging very low, but it gets lower and lower as time goes on, so lower with advancing age, which is exactly the signature, the genetic signature of programmed death. Point 4, and point 5, the last one. The classic theory that opposes programmed aging is that, well, you know, maybe there are aging genes, but there are pleiotropic effects. In other words, these genes aren't there because they cause aging. They're fertility genes that happen to have a side effect, and the side effect causes aging. But they're there primarily for fertility. That idea is due to Williams in 1957, and it was brilliant. I mean, it was exactly the right thing to see in 1957. Yeah. Of course, in 1957, people didn't even know what a gene was, really. There was certainly no means of isolating genes and seeing whether this is true, but it was a brilliant hypothesis, and it was put right out there. And now we have the gene. And it turns out that some of them have to do with fertility, but a majority of them don't. Especially these high-level genes I talked about that are common to many animal species and even to single-celled entities, they don't seem to have much to do with fertility at all. So those are my 5 points.
The 5 points, yeah. Well, you know, I think we can get into a couple of those, and I want to, again, remind everyone, if you had any thoughts or questions on those 5 points, you could please ask them in the chat. And I guess the one I want to get into is the genes, the high-level genes for aging. And would you call telomeres one of those high-level regulators of aging?
Absolutely. And, you know, telomeres control aging in protists, in these tiny one-celled organisms. Not bacteria, but protists or eukaryotes like us. They're much larger cells. A little background about what a telomere is that appropriate?
No, I think we're good. I think everybody knows. Yeah, most of the audience does know about telomeres. Now, I've been in on this argument before, and I want to hear your perspective on this. In that some people have argued that some cells in the human body don't divide and therefore are not subject to that master regulator. Also, human beings typically don't live long enough to go through enough cell divisions in order to die from telomere shortening. So those are 2 arguments I've heard, but you can certainly correct me in that thinking. But those are 2 arguments I've heard why at least that one master regulator of aging perhaps is not does not play a big part in humans.
Well, clearly there's a lot of work that needs to be done on the details of this. And people who say that the cells don't divide enough times to make telomeres relevant, and that's a theoretical position, and it's becoming less and less tenable as we get evidence in. But I admit the evidence is rather thin at this point, but I'll get to what it is. Let's start with telomeres in protists, in protozoans.
Okay.
Telomeres are the primary method of aging in protozoans. It's like a clock. every time the cell divides, the telomere gets a little bit shorter, and when it gets too short, you can't go on. I mean, it's clearly a mechanism. There's no wearing out in this at all.
For single cells.
For single cells.
Organisms.
That's right. And the mechanism for resetting the telomere clock is sex, or conjugation it's called in these one-cell animals, and that resets the telomere clock. The enzyme telomerase, which extends telomeres, is part of the genome. It could be expressed anytime for any reason. It's free. There is no cost to extending the telomeres, but the cell withholds the enzyme until it's conjugating. And the only reason for this, there are no other hypotheses put forth. The only conceivable reason for this in protists is that it's a programmed mechanism of aging. Now, people have known this for about a generation now, but everybody assumed, as you said, well, this couldn't possibly have to do with aging in mammals. It's much too simple.
Sure.
If you look at mammals, the theory was, well, we have telomeres that are long enough that it doesn't affect our lifespan. There was a study, I think it was 2002, out of Utah, a retrospective study with blood that had been kept in hospitals 30 years earlier. They compared, this is Richard Cawthorn.
Okay.
He compared the length of telomeres in these 30-year-old samples of blood with what happened to people in the intervening time. What was their life expectancy? They were middle-aged when the blood samples were taken. They were in their 50s. And here, 30 years later, was there any relationship? And he found a striking relationship. There was a factor of 2 difference between the mortality rates in the highest quartile of long telomeres and the lowest quartile of short telomeres. A factor of 2 difference in the mortality rate. This is tremendous. I mean, you've got to think that the telomeres are having some effect in humans. Now, this mantle has been picked up by the lab of Elizabeth Blackburn out at University of California, San Francisco. And she, of course, discovered telomeres and their function, 30 years ago. And she's still very much an active researcher, a big brain, perennial candidate for the Nobel Prize. And she's now doing studies of humans, measuring telomerase expression, measuring telomere length, and again, people's life expectancies, and trying to go after the mechanism.
And basically, the shorter the telomere, the lower the life expectancy.
Well, that's right. The shorter the telomere, the longer you live. And the link is especially true for heart disease. The rate of heart attacks is dramatically lower in people with long telomeres.
Okay, what about this thought here that a viewer just brought in, is about the relationship between cancer and telomerase, and that it's not an aging clock. Instead, telomerase is not expressed in order to prevent cancer in an advanced age organism. So cancer prevention is the primary reason, and that aging is just a side effect of turning off telomerase and allowing telomeres to shorten.
Well, absolutely, that's the standard theory.
Okay, and that's standard, yeah.
I would be skating on very thin ice if I poo-pooed it.
Oh, okay.
But let me say, it was put out as a theory before anybody had any evidence.
Okay. So this is one of those that
absolutely.
It's an evolving kind of theory now, as more evidence comes out, you would say.
Well, if you suppose you believe that programmed aging is impossible, as most evolutionists do, they believe for theoretical reasons that programmed aging is impossible. They think they can prove it. I'm more of an empiricist than that.